Thank you for joining me for the 7th episode of the Doc Talk with Liz podcast. Today's episode features Dr. George Church. Dr. Church is a geneticist, molecular engineer, and chemist, and is a professor and researcher at Harvard and MIT. I hope you enjoy. Hello, Dr. Church. Thank you so much for joining me today.
It's great to be here. Thank you.
Can you kind of start off just by telling us about your journey into genetics, synthetic biology? I know that your research kind of goes across a few different types of fields. So if you could kind of tell us about that.
Yeah, I guess I got interested in almost all branches of science pretty early on. I couldn't decide which one was most interesting, and I'm 66 years old and I still can't decide which one is most interesting. I think they're all interesting, all of STEM. It started as a kid. I would hang out on the swamps, or the bay,
Yeah.
and we were close to the water, and I would just see what kind of interesting things floated up or were in the mud. Snakes and echinoderms and things. And then I was adopted by a physician, and I got to know technology. So there was one was the very, very kind of mucky natural world, and the other was this beautiful stainless steel, sterile,
Right.
Pharmaceutical. He did house calls, and that was all fascinating. And then I also got exposed to computers in the form of animatronic robots, like Abraham Lincoln, that at the World's Fair when I was 10 years old. So I wanted something that included all of these three things and more, and I finally found that, or initially found that, I guess, with X-ray crystallography as a sophomore in college. And it was just great because you had to use all of these ideas in one place, and I haven't turned back since. I mean, I've added a lot of things other than crystallography, like reading and writing DNA, RNA, protein, cells, ecosystems, but they're basically all the same kind of thing where you need to have an interdisciplinary focus. Now that's not for everybody, but that happened to work for me.
Okay.
Yeah.
Gotcha. Awesome. And so where did you go to your undergraduate? What did you major in, and what research did you do as an undergrad?
Yeah, so I took advanced placement exams because I didn't want to repeat what I had learned in high school at all, and I did much better in physics and math than I did in chemistry and biology, but I wanted to major in chemistry and biology.
Okay.
And so there was one school, Duke, that would accept my poor AP scores to allow me to pass out of the first year. And so I went to Duke. I did get 2 degrees in 2 years, zoology and chemistry, and then continued at Duke as a graduate student. First in microbiology, and then I transferred to biochemistry, and then I flunked out of biochemistry. So it was a very whirlwind tour through Duke, where I went through 4 departments in about 3 years.
Wow.
Finally failing because I was spending too much time in the lab, which is your second half of your question, was the research. So as before I got to graduate school, I did undergraduate research on mycoplasma, mostly during the summer at the Boston Channing Lab, Harvard microbiology lab. And then when I came back to Duke, I was studying virology, not doing experiments, but I was really doing this X-ray crystallography on RNA with Sung-Ho Kim, who was a wonderful advisor. Both for me as an undergraduate, as a graduate student, and then as a technician after I flunked out. and he helped me get back on my feet and get back into a PhD program at Harvard.
Right. Okay. Awesome. Gotcha. So then how does that kind of connect to the current research you do? And can you kind of tell us, I know that your research now, once again, just spans a lot of different fields and you're looking at a lot of different things. So can you kind of talk about some of that, like what you're currently researching and how that kind of relates to what you researched in your undergrad?
Right. So doing X-ray crystallography opened up a lot of doors. It was really the first field, almost one of the first field in biology or chemistry that had computers as a key component, had robots. I mean, there was a robot in a cold room that would just keep collecting data, like moving in these arcs, collecting X-ray data, X-ray diffraction data. And that I got, but there was very little computing or robotics in most of biology. So I said, well, I just started applying what I learned in crystallography to DNA sequencing, and then to protein sequencing, and then to RNA. And these were quantitative, how much protein and how much RNA, not just their sequence.
Right.
And that led to functional biology, which led to systems biology, which led to synthetic biology. And now I would say everything is kind of unified under synthetic biology, which is, can you make it? And ability to make things allows you to test hypotheses, kind of in the classic bio research, but it also allows you to do gene therapy, it allows you to do genome editing, new genetic codes, all kinds of things. So synthetic biology probably is the unifying theme right now. But you can see how we got there through this series of DNA, protein, RNA, functional genomics, systems biology, and then synthetic biology.
Okay, gotcha. Do you possibly have like a favorite like protein or something, or like a favorite kind of concept in science that you could kind of explain to people?
Sure.
That you think, yeah.
I have kind of two favorites. The one that's been with me the longest is the genetic code and tRNA. So that was that first molecule that, as a teenager, in college, I got into crystallography of tRNA, and so we still work on tRNA, 40 some years later. So that's a question, a molecule, a favorite question, a favorite molecule, a favorite approach, a methodological approach is, just a little bit more on that molecule. That the process of protein synthesis is one of the oldest and most conserved processes in all of biology. The genetic code is pretty well conserved in all organisms. The most conserved molecule sequence in the world is the ribosomal RNA, and the 3D structure of the tRNA, which is what I worked on. Those are they're everywhere. Okay, so that's partial justification. It's the name central dogma applies to it. So that's what I've been working on. Okay, but the methodological thing I think that really has made our lab very successful, that most people don't even realize what the secret sauce. It's not secret. It's perfectly, it's in all of our papers, but they don't see it. Is called multiplexing, or molecular multiplexing, and that is, people get it confused with parallelism. So it's like you can make your lab faster and more productive by buying lots of the same instrument and just filling your lab with this instrument and just keeping it full. But it doesn't actually save you any money, because the more instruments you have, the more money you're spending, right? But what multiplexing does is, given an instrument, something as simple as a pipette device, or something as complicated as a sequencing device, each droplet, instead of doing one reaction, can do billions of reactions. The same droplet, the same amount of chemicals, the same amount of equipment time. And the idea is each molecule has, or each molecular type has a barcode tag, just like the barcodes in the supermarket, but this is now molecular barcode. And that allows you to mix them all together, randomly, and then do many, many steps. So, and then then you can analyze them by the barcodes. And so we do multiplex sequencing, multiplex synthesis, multiplex protein evolution, developmental biology, where the cells are barcoded and so forth. So it's a general method, and it's very different from automation or parallelism. I mean, it can include automation and parallelism, but automation and parallelism don't do nearly as much, and they're not nearly as flexible. And so multiplexing, I think, has, for example, brought down the cost of sequencing by 20 million fold. 20 million fold.
Wow.
Imagine that your clothes and your house and your car were all 20 million times cheaper. I mean,
That would be awesome.
it would be nice. So anyway, DNA sequencing is 20 million times easier, and writing DNA is also similar amount.
Mm-hmm. Wow, very. In the first part of this podcast interview, Dr. Church mentions that he has been interested in all areas of STEM from a young age and incorporates many aspects of STEM into his current research. He also mentions that he went to Duke for his undergraduate and graduated with 2 degrees in 2 years. As a kid, Dr. Church would hang out by the bay and investigate the different things that would come up from the water. He was adopted by a physician, which introduced him to more aspects of STEM. Dr. Church studied X-ray crystallography in his undergrad. This allows us to determine the atomic structures of proteins. Dr. Church's favorite molecule is tRNA, which is transfer RNA. The 3D structure of transfer RNA is very well conserved. His favorite approach is molecular multiplexing, which allows you to do billions of reactions at once with a molecular barcode. And molecular multiplexing has actually brought down the cost of DNA sequencing by 20 million fold. It has also brought down the cost of writing DNA by about that much. Very cool. Okay, so now I have another question, and I don't know if you'll be able to give me a clear-cut answer on this, but do you prefer human genetics or bacterial genetics?
Yeah, that's a tough one. I mean, we do both, and
Right.
I would say we do probably similar amounts. Now, human, I'm going to generalize to mammalian because we work on humans, dogs, elephants, and let's see, elephants and mice. Those, and to some extent, they're interoperable, but they have different goals. So the elephants, we're trying to save them from as an endangered species and to help us sequester carbon in the Arctic. For the dogs, we're trying to reverse aging, both for pets and also as a way to get to humans, many human diseases which are age-related. Mm-hmm. Mice are usually just a way of testing things quickly relative to dogs and elephants and humans. And then bacteria, they have their own utility, I mean, which is that they're used for manufacturing. They're grow faster. So we have bacteria that will double in 15 minutes. So these are even faster than E. coli, which is the workhorse, which doubles in about half an hour. These Vibrio will double twice as fast, and it means that I mean, it's just astounding to see how fast these things replicate. So there's advantages to each. I would say our lab is slightly biased towards the mammals.
Okay.
Because that's what we are, you know?
Right. That's true. So, awesome. So you mentioned reversing aging. What causes aging in the first place?
Well, aging is almost certainly programmed into our bodies from birth. Some of the evidence that it's programmed, that it's determined, it's like planned obsolescence is a term in manufacturing of goods. Like your foods have an expiration date on them. So mice have an expiration date of 2 years, and bowhead whales have an expiration date of 200 years, and humans are somewhere in between.
Right.
And so that's the other way we know that it's programmed. And so it's programmed genetically and epigenetically. Epigenetically is basically how the genes do their thing, how they play out. So you can think of the genetic as kind of like your information on the disk, and the epigenetics is how that information on the disk turns into, sound on your speakers and images on your screen. So we have really good control over epigenetics at this point. It's part of the technologies that we develop, like reading and writing DNA. That's also regulating DNA function.
Mm-hmm. Gotcha. Awesome. And then can you also kind of give us kind of, I always say, a glimpse into the life of a synthetic biologist, but kind of like, what's your day-to-day? Kind of like, what do you do? What do you do?
Well, it depends on which synthetic biologist. So just whether you're running a lab or whether you just started in the lab. But I would say my day, pretty much ever since I started doing synthetic biology, or actually even since I started doing science, is a mixture of doing computing and doing bench work and thinking and talking about doing computing and doing bench work. So people sometimes neglect the thinking and talking, but it's a huge fraction of your day. Even if you're, quote, working alone, all by you shouldn't work alone, but even if you think that's what you're doing, you still spend a lot of time planning the experiments, writing down what you're going to do, and then taking apart to figure out what happened, what went right and what went wrong, and drawing conclusions. So that's a huge fraction of the day. And it's actually quite pleasant, I think, talking to people about their experiments, about your own ideas as well. But that's a big part. You can work by yourself. It's fine. In fact, there are many ways to succeed in science. There's no one way. You can be a loner. You can be very gregarious. You can like math. You don't have to like math. You can like working at the bench with things that are very messy or things that are very clean. There's a lot of personality types that are very helpful. And indeed, some of the people that normally wouldn't fit in do fit in, because in science, diversity is incredibly important.
Right. Mental
diversity, flexibility in some cases, rigidity in other cases, where you just keep going in a particular path and nothing stops you for decades. But other people, what they're good at is jumping around and finding connections.
Mm-hmm. Yeah. Absolutely. And then what do you like to do in your free time?
Yeah, so free time,
you have
I do have free time.
Okay.
It's from the outside, it may not look that way, having started 30 companies and working with 30 companies I started, right. And teaching and talking to Congress and so forth. But I have a fair amount of free time. It's mostly spent learning what other people, in particular my daughter and her daughters, like. So by seeing what they like and helping them do what they like, I learn a tremendous number of things that I would otherwise never be exposed to, like fashion design and balancing and all sorts of interesting activities. So that's what I mostly do in my spare time, is hang out with my daughter and her daughters.
Awesome. That's great. Family time is definitely something that I enjoy myself as well. But so you talked a little bit about the science so far. So how about, I'm kind of interested in like the ethics of this. Like one thing that I've learned through the iGEM competition is like always considering human practices and stuff in a project design. So how do you kind of incorporate that into your project design?
Well, actually, so our lab is probably at the extreme edge of the technology development world, both in terms of we do very cutting edge technology development and our cutting edge ethics and safety. And we I don't know, I've done about 20 some peer-reviewed papers on ethics and safety. And I consider it part of engineering. Most engineering disciplines have a sub-discipline which is about ethics and safety. You know, if you build a bridge, you don't want it to fall down with cars on it. Also a component of ethics is communication with the public. You can't just say, "Oh, I know what's right, and I'm going to enforce it." Right. It's more a matter of finding out what people actually want, which changes from person to person, day to day, culture, different cultures, and so getting consensus is challenging. But sometimes there's a win-win where it's not 49% lose and 51% win, which I think is not a good political system, but it's one we're very familiar with. It is more a matter of finding where both can win. You know, so rather than some of us who don't want to give up our SUVs in order to solve the climate crisis, come up with a way of doing carbon sequestration, which is so cheap and basically natural. Nature takes care of it, but you just needed to give it a little nudge. Right. Then that's a win-win. Then people can have twice as many SUVs if they want, right? If it's really successful. But you don't put the cart ahead of the horse. You don't say, "Oh yeah, it's obviously going to be successful. Let's just start burning fossil fuels again."
Right. Yeah.
So always looking for that win-win is helpful. And equitable distribution is a key component of the ethics. We don't want to create technologies that are only available to the extremely wealthy. Now that was somewhat the case for cell phones. Back in the day when we weren't sure whether cell phones were good for you, they were going to melt your brain. They were very powerful and mysterious, and people thought that putting this against your head would be dangerous. But they were also only available to the wealthy, and they were large and clunky. But by the time they got to be affordable and safe, they're widespread. But they're not fully widespread. Not everybody has full access to the internet and to calls. An example of technology that is widespread are certain infectious diseases, which have been smallpox, guinea worm, polio, leprosy. These are extinct or almost extinct, and that means that nobody has to pay even a penny for a drug or a vaccine. Right. Now, vaccines are pretty cost effective, but extinction's even cheaper. If you make the pathogen extinct, you don't no longer have to manufacture the vaccine. So,
right.
Anyway, those are examples, and I mentioned earlier bringing down the price of sequencing by 20 million fold. My wife and I do a kind of an ethics, it started an ethics movement, a couple of them, PG Ed and PGP, and we do congressional briefings and talk to screenwriters and provide high school materials online.
Oh, awesome.
Among other things.
Okay.
So you need to get the word out, because the average person, you can give lip service and say, "I want to engage everybody," but most people don't know that you want to engage them, and many of them don't care, and so you have to make extra effort to make sure everybody, or a lot of people, are engaged. You won't get everybody.
Right. Yeah, definitely. I'll do my best to kind of spread that around and tell my friends and tell my friends to tell their friends, and so
that's how it works.
Yes.
And you're part of the solution, definitely.
Mm-hmm.
Yeah.
Okay, so now you kind of talked about the ethics, the science, kind of what you do. Now, do you have any tips for people that want to go into synthetic biology or science, or kind of do some stuff like you do?
Yeah, well, for one thing is you want to pick a field that has a future. And one of the ways that you can tell that is a field that is constantly leveling the playing field frequently. So every couple of years, so reading and writing DNA and allows you to read and write all kinds of things, both biological things and even non-biological things like information and materials that are non-biological. But anyway, those are going exponential, and that exponential is a series of innovations of little methods along the way. And every time a new method kicks in, the playing field is level again. Everybody has the same, more or less, closer to the same opportunity of getting it on the ground floor. But in other fields are very static, and they have hierarchies, and they have people that are protecting the status quo. And you want to be very cautious before you move into one of those fields.
Right.
But something that's constantly under chaos and mixing and stirring up disruption, you should also be cautious there. But there's something interesting going on there, and you've got a chance of getting on the ground floor, even when you're young and don't have much of a track record.
Right. Yep. Definitely. Okay. And so you kind of talked to us about like tips, and you told us some interesting science concepts earlier, but do you have some more interesting science facts that you could share for us?
Yeah, I mean, I think you have to find things that are not only interesting to you, but interesting to the world. And very often that's another case of a win-win, where you can find that what you're addicted to, the world wants you to do. I mean, there's certain addictions which the world definitely doesn't want you to do because they disable you basically. But there's others that are enabling, and a lot. So for example, some of the things that we do, a mammal that I didn't mention earlier is pig. So we work on pigs. We've made them, humanize them so that they can donate organs to humans. And that's I'm a vegan, and so I'm not that big a fan of eating pigs. But that's 3 billion pigs a year, while the number of pig organs that you would need to solve the transplantation crisis is just a small number. So this is where your personal preferences come in. Do you want to work on behalf of animals, like aging reversal in dogs, and saving endangered elephants from herpes virus and giving them new land to grow in far from humans? Or do you want to use animals as a way of helping humans? That's a personal preference, and you need to think about what your personal preferences are. Or do you want to work with something cleaner, like chemicals or instruments? So it's good to try a bunch of things out, because sometimes things that look good on paper, once you get really close up to them, you realize that, microscopy seems like it's very clean and easy, has good hours and so forth, and increasingly automated. But at least in the old days, microscopy wasn't so good for some people in practice, because you'd be sitting all day looking through a microscope, not making eye contact with other people and so on. Now that's changed. Now it's much more automated. But pay attention to where things are going too. Try to extrapolate that curve, because by the time you get trained, the world's going to be quite different, and so you need to anticipate what it's going to be like rather than what it was last year.
Right. And so you said, like, it's important, finding a win-win. So, something that you're addicted to and the world actually wants you to do it. So what are you addicted to? I can ask, kind of.
Well, I'm addicted to all kinds of science. I'm addicted to interdisciplinary science, and so that's mostly the world wants specialists, and I think that's very valuable. And there are a lot more niches. It's like, but every now and then you need to connect. Sometimes you need to connect the different disciplines, and you need somebody that will do systems integration, for example, that'll put, instead of developing a new transistor or a new pixel on your screen, puts together a whole computer system or a whole, home electronic system, right? And so you have to decide whether you're a generalist or so in my case, I'm a generalist, and that fills a niche in society. What else am I addicted to? I'm addicted to, I guess, fact finding. It's like truth, kind of for its own sake. And so whenever anybody, and this has actually been transmitted to my daughter and via both my wife and I, and to the grandchildren, is that somebody will say something and they'll just like check it out on Google, see if it's really, get a little more structure to it. So rather than accepting things as truth, you question authority. So I'm a little addicted to questioning authority, including my own authority and people. So I think that's a good one to be addicted to, is to make sure you got your facts straight, make sure everybody else has their facts straight, and it's easier than it's ever been. I mean, it used to be checking out one fact that you might say would take me all day in the library looking through books and finding stuff. Now it's a simple, you don't even have to read, you just talk and it shows you images. My little 2-year-old Nova granddaughter, when she was 2, she was addicted to asking questions. Even though she couldn't read, it was like she would ask a question, and you could get her to. So these are good addictions.
Yeah, definitely. Well, it looks like it's about 11, so I want to just say thank you so much for joining me today, and thank you for your time. I had a really fun time with this interview.
Yep, same here, and I think what you're doing is great. You're, like I say, part of the solution, and keep up the good work, and hopefully many people will be inspired by you and do similar things. So
thank you.
In the last part of this podcast interview, Dr. Church mentions that he works with both bacterial and mammalian genetics, but prefers mammalian since that's what we are. Dr. Church and his wife started PGY and PGP, which are movements for ethics. It's important to find a win-win and focus on equitable distribution when dealing with ethics. Genetics is like the information on a disc, where epigenetics is the sound that comes from the information in the disc. Dr. Church's day consists of computing, sometimes bench work, and talking with other people. He also mentions that there are many different ways to succeed in science, so it's important to find what works best for you. In his free time, Dr. Church likes to learn what his daughter and grandchildren like. This teaches him about things that he doesn't know about. Lastly, Dr. Church mentions that it's important to focus on getting into a field that allows you to get in on the ground floor. And also, it's important to find things that the world wants you to do, and along with that, to find your personal preferences. Thank you for watching this episode of the Doc Talk with Liz podcast. Do not forget to like and subscribe for future videos.